Hi, good morning. This is Mscdfsm Biochemistry. I'm Kavita Kundra from Kavita Kundra Wellness Studio, and we are here with our vitamins and minerals second part. Vitamin E is generic name for two families of compounds called tocopherols and tocotrienols. What are the functions of vitamin E? Vitamin E is a natural antioxidant. Vitamin E is fat soluble. It accumulates in lipoprotein circulating in blood cell membranes and fat deposits. Without vitamin E, cell membrane, active sites, and DNA are less protected from free radical damage. PUFA present in the body can readily undergoing peroxidation by oxygen due to the presence of double bond. These peroxides undergo further series of reactions to generate free radicals. Lipid peroxidation is a chain reaction providing a continuous supply of free radicals that initiate further peroxidation. The free radicals are extremely toxic to our tissues. Vitamin E is an antioxidant. It acts as chain breaker and trap free radical in cell membrane and plasma lipoprotein. It acts as lipid peroxide radicals formed where there is oxidation of double bond present in PUFA. This prevents chain reactions. The product formed between tocopherol and free radical is relatively unreactive. It forms non-radical compounds. In this way, free radicals are prevented from causing an injury to cell. Vitamin E molecule gets converted to tocopherol oxyl free radical product in which vitamin E is an oxidized form. Vitamin E, the tocopheroxyl radical, is reduced back to tocopherol by reaction with vitamin C from plasma. This vitamin C is generated to continue its antioxidant functions. When vitamin C reacts with vitamin E free radical complex, the free radical transferred to vitamin C forming vitamin C free radical complex called monodehydroascorbate free radical. In this process, vitamin E gets regenerated. The monodehydroascorbate free radical then undergoes enzymatic and non-enzymatic reactions forming ascorbic acid and dehydroascorbic acid. They are not free radical, hence they are not toxic. Vitamin E is a scavenger and protects unsaturated fatty acid from peroxidation reaction. Tocotrienol occur only at low level in nature. Tocotrienol are highly potential from tocopherols. Tocotrienols reduce cholesterol level and have antithrombic effect and prevent cardiovascular disease. They also have anti-cancer effect. So this is the cycle. You can go through the cycle: vitamin E cycle, vitamin C, thiol cycle, and then this is an easy cycle, not the complex. So vitamin K, what are the functions of vitamin K? Vitamin K is required to form a blood clot. It is required for the synthesis of protein which are needed for the process of blood clotting. The proteins needed for blood clotting are secreted in inactive form called precursors. Vitamin is required to make them in active form. If vitamin K is low, then it results in decreased clotting of the blood. Prothrombin is a blood clotting factor 2. Prothrombin is synthesized as an inactive precursor called pre-prothrombin. This is a protein molecule and it contains the amino acid glutamic acid in the polypeptide chain. Conversion of pre-prothrombin to thrombin requires carboxylation of some glutamate residues. This is catalyzed by carboxylase enzyme which requires vitamin K for its activity. The gamma carbon of glutamate residues is carboxylated to form gamma carboxyglutamate. Gamma carboxyglutamate residues are good chelators. These are organic compounds which have the ability to bind metal ions. In this case, they bind to calcium ions. The prothrombin calcium complex formed converted to thrombin. Thrombin converts the plasma protein fibrinogen to fibrin. Fibrin has mesh-like structure and traps red and white blood cells and other plasma protein to form blood clot. Vitamin K. Vitamin K cycle depicts the carboxylation reaction. Vitamin K epoxide is recycled to vitamin D hydroquinone by enzymatic reduction. In epoxide, two carbons share an oxygen atom. Furthermore, two hydroxyl group get converted to double bonded oxygen. For continuous blood clotting, hydroquinone form must be regenerated. Reduction result in loss of epoxide oxygen forming quinine structure that retains in the loss of epoxide oxygen forming the quinine structure that retains again two double bond oxygen atom. This reaction catalyzed by vitamin epoxide reductase and neat compound with 2SH group. Hydrogen ion are added to two carbon that have an epoxide oxygen. Now vitamin K undergo second reduction reaction catalyzed by vitamin K quinone reductase and vitamin K hydroquinone is regenerated. This reduction required the participation again of sulfur hydryl compound to donate H plus ion for forming the O groups and cyclic process continues. Vitamin K is also needed for synthesis of two calcium binding protein named osteocalcin and bone matrix protein. Both contain glutamate residues that has to be carboxylated to form gamma carboxyglutamate residues in surface of vitamin K so that calcium get deposited in the bones. Water soluble vitamins. These are the soluble in water. Fat soluble vitamin gets stored in the liver and hence if they get accumulated it leads to the toxic effect. But these water soluble vitamin when get in excess they get excreted in urine. Fat soluble vitamin exert a different type of effect while a large number of water soluble vitamin like the so-called vitamin B complex vitamin have one type of activity functioning as coenzyme in the body. Coenzyme are non-protein molecules that are required for activity of many enzymes. What are the classification? One is energy releasing water soluble vitamins like vitamin B1, B2, B6. Homeopathic water soluble vitamin and other water soluble vitamin like ascorbic. Energy releasing water soluble vitamin. These vitamins act as coenzyme. They get absorbed in intestine and each vitamin undergo modification and converted to form that takes part in the reaction. These active forms get synthesized in liver. These 6 vitamins have similar functions and their deficiencies lead to similar deficiencies. In many cases, the nervous tissue is involved due to higher energy demand. Thymine, vitamin B1. Vitamin has central role in energy yielding reaction of carbohydrate metabolism. The active form in which thymine functions in the body is called thymine pyrophosphate, that is TPP. Structure of thymine. This is the simple structure of thymine: aminopyridinium ring, thiazolium ring and 2-phosphate. Functions of thymine. TDP is the coenzyme for 3 multi-enzyme complexes that catalyze oxidative decarboxylation reaction. These are pyruvate dehydrogenase in carbohydrate metabolism, alpha ketoglutarate dehydrogenase in the metabolism of branched chain amino acid, that is leucine, isoleucine and valine. It converts alpha keto branched chain acids obtained when the branched chain amino acid undergo the transamination to corresponding enzyme derivatives. It is known as the enzyme of multi-step pathway, function as a group and within complex it is integrated by various required coenzyme. Pyruvate dehydrogenase complex consists of 3 enzymes working sequentially. These are pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase along with 5 coenzyme. These are TDP, lipoic acid, coenzyme A, FAD, NAD+. The final product is NADH is formed, which is oxidized in the mitochondrial electron transport with the formation of energy rich ATP. So this is it, and we will be continuing again with another topic of vitamins and minerals. And please share it with your friends who needs these notes in the corona time, because classes are not going on, so we can help them out. And please like, subscribe and share. Thank you so much. Stay safe, stay protected.